going to talk about three scenarios, three different projects from the lab that I think will tell us a little bit about how someone like me, who works primarily in basic science, look at the pathophysiology of Alzheimer's disease. So Alzheimer pathology, so far at least I haven't heard too many people talk about this in the talks yesterday, and basically it has a traditional senile amyloid plaque. It also has neurofibrillation angles. The plaques are extracellular, tangles composed of tau is intracellular. There's obviously neuronal loss. There's also a synapse loss. And then there's finally various amounts of inflammatory responses in the brain. And so here is on the left is an example of the neurofibrillation angles using a silver stain, as was seen initially by actually both Cajal before Alzheimer himself. And then by electron microscopy, these are so-called paired helical filaments that's inside the cell body of these neurons. And then here, using an antibody to stain the amyloid plaques, they can be substantially quite large, and they're extracellular. And right here, colored in brown and in blue, if you can make it out, are the cell bodies of neurons that's in the brain. And then schematically, this is what we see. We have the tangles in the neurons, and the tangles could also be in the distro neurites of the neurons, extracellular amyloid, and then reactive astrocytes and reactive microglia. Okay, so this is a rather prophetic statement from Dennis Selkoe from 2002, and actually he turns out to be quite correct, which is that the underlying pathophysiology of Alzheimer's disease that leads to cognitive impairment is in fact a dysfunction at the level of synapses. And loss of synapse has been shown by a number of laboratories is the major correlate to cognitive impairment in individuals affected by this disorder. And here we have traditionally emphasized neuronal loss. Here in the cholinergic nuclei, the nucleus basalis of Minard, in AD brain, you can appreciate the profound neuronal loss in the cholinergic neurons versus an age-matched control. This is actually the original case that was published by Peter Whitehouse and Don Price almost 25 years ago that led to all the cholinergic therapy for AD. But if you look more carefully, if you look more carefully at synapses using synaptophysin, this is an antibody of synaptophysin, instead of cell loss, you will also see about a 50% drop in synapses in brain. And this is work from Bob Terry and Esli Azamazlia from my institution in the US at the University of California, San Diego. I'm going to tell you three stories now, and if you fall asleep, these are the three major conclusions that I will hopefully guide you through. One is that in APP gene-targeted mice, I'll come to explain it, APP is the amyloid precursor protein, which is a precursor to the amyloid, the A-beta, that's in these amyloid plaques. A-beta toxicity, or the amyloid toxicity, progresses anterogradially. It goes from the cell body to distally, presumably by the release of A-beta from presynaptic neurons to impair function of postsynaptic neurons, okay? And I'll show you the data to support this conclusion. Second is that in tau, instead of APP, in tau gene-targeted mice, a similar toxicity on postsynaptic neurons like A-beta, but neurons expressing tau also exhibit impaired synaptic function. So tau toxicity seems to be more severe than A-beta toxicity. And finally, one pathway of A-beta toxicity that we've been studying in the lab for more than 10 years is APP dependent and appears to involve caspase-mediated cleavage of APP through localized caspase activation. A long sentence, but basically we believe that there's localized damage to dendrites, and that's responsible for causing the synapses to go away without killing the neurons themselves. Okay, vignette one, targeted expression. So here, Alzheimer's therapeutics in rodent models. So the good news, there is a lot of good news from this, if you study rats and mice, is that there are many ways to treat Alzheimer's in mice successfully. Okay, The disconcerting news, that is not so good news, is that there are more than 100 different ways to successfully treat AD, Alzheimer's, in animals. Okay. The really bad news, however, is that none have worked in humans. So there's something about the rodent models that we study that ultimately does not quite translate to useful, effective therapies in human. And the question is why? So here's a transgenic example of a transgenic mouse model. So all you have to appreciate is that this is a cross-section of a mouse brain, and each black dot is one of the amyloid deposits. So we can generate mice with a ton of amyloid deposits in brain. That's not the problem. The problem really is virtually all rodent models overexpress APP. Therefore, not only is there amyloid effect, there is just a simple fact that these animals overexpress human APP gene at supraphysiologic levels. That isn't physiologic, okay? That's number one problem. Most models show little or no cell loss. So even with that amount of amyloid in brain that you can see here, there's very little cell loss. The neurons don't die. And then not one single disease-associated mutation in humans can drive the full spectrum of AD pathology in mice, especially tau pathology. So in fact, we do not generate a mouse using any human gene mutation discovered in humans that really can be replicated in a single mouse model. That doesn't happen, unfortunately. And so we're not able, therefore, to link the two major pathology together in a mouse. That is the amyloid and tau pathology. It requires multiple genes and you put them together in a mouse, which by definition is therefore not physiologic. Okay? So my good friend Dale Bredesen at the Buck Center in Northern California likes to call this the Mouseheimer rodent model because it really isn't an Alzheimer model. They all replicate only certain aspects of AD pathology. And so the first generation of AD mice, or APP mice, is just overexpression. They're not selective. Second generation is neuronal promoter, so at least we could target the transgene only to the neurons. Third generation is conditional expression of APP. That's regulated by TTA, that is the tetracycline transactivator. So basically, tetracycline would control the expression of your transgene, and then depending on what directs the TTA expression, you can now generate mice which only certain populations of neurons are affected, and I will show you that momentarily. And in the fourth generation, our humanized A-beta, that has no overexpression, is only endogenous. I'm not going to talk about that. That came out only recently from Takaomi Sato's lab in the Riken Institute in Japan. So I'm going to tell you about a variation of generation 3 that our lab have been working on for a while. So in this particular case, we need to put 3 genes together in a mouse. Again, very non-physiologic, but that's the only way where we could site-specifically express APP in different neurons in the hippocampus. In this particular case, by combining 3 genes, the GERK4, which directs, in this case, the Cre recombination expression in the hippocampus, together with the TTA CUSAT. That TTA by itself is quiet, but it is excised by the Cre recombinase, and then the TTA is liberated to drive the APP transgene. So 3 of them. So by Cre cutting the lock-stop locks to liberate the TTA, and then now the TTA will turn around and then bind to APP, and then we have APP expression. A little bit complicated, but that way we can target the APP transgene only to neurons in the hippocampus, and particularly in CA3. And in this case, we're able to, using different Cre promoters, POMC, GERK, and CAM kinase, we can direct expression to dentate, CA3, CA3, I want to go back here because you can see that it's only in the dentate in brown here. You can see that it's in CA3 very nicely in brown, and then finally in CA1. It's going too fast, too sensitive. But that because of the TTA expression, the lovely thing about the TTA system is that if you add doxycycline, this is a so-called TET-off system, By adding TTA into the system, then TTA now blocks expression and you turn the gene expression off. In this particular case, this is what we get. If you stain immunocytochemistry, here's a non-transgenic mouse, here's a transgenic mouse. It's quite easier to see this immunofluorescence image. You add doxycycline for 2 weeks and the expression goes away. So this shows you that we can target the APP transgene only to CA3 neurons in this particular picture. So what happens when you do that? And what happens is that here you can see lots of amyloid deposits, just like I showed you earlier in brown. And then now here is CA3, okay, and then here's CA1. So in CA1 animals, the deposits are all out here, which is the so-called entorhinal cortex. And that's where the axons coming out of CA1 neurons terminate. Here's an example from CA3 mice coming out from the CA3 neurons in brown, and the amyloid terminal plaques are here in the CA1 region, and that's the axonal terminals of the CA3 mice. So this shows that the amyloid are abundant, but that they're only present in the axonal terminal field. So this means that there is an anterograde or something coming out of the neurons that's deposited in the axonal terminals. So what about electrophysiology? What happens in these animals? Ultimately, synapses work by neurotransmission. So if you measure a long-term potentiation as a measure of synaptic plasticity, here using CA3 mice, so the expression here in green is in the CA3 area, you stimulate the Schaefer collateral, record in the CA1 neuron. Remember, CA1 neurons is where the amyloid deposits are seen. Then you can see that very nicely. There's depression of LTP in the transgenic animals, okay? It's been shown by many labs that these APP transgenic mice would show synaptic depression, so we can see it in our mice. But what's interesting is that same animals, but if you stimulate the incoming fiber, so these are now coming from the dentate gyrus to CA3, so these are coming from neurons that do not express the APP transgene. It's being expressed in the postsynaptic neuron. Then in that case, there is no depression of LTP that we saw here in the earlier slide. Oh shoot. Okay, in the earlier slide. We can only see it, we do not see it when the postsynaptic neuron is expressing the transgene. So here, summarizing what I just told you, which is that now if you look at CA3 mice, you simulate the outflow tracks, the axons, you see impairment. You simulate the inflow, you do not see any impairment, okay? Now, if you look at CA1 mice, I told you we can express it in different neurons. You express it in CA1 mice, then if you stimulate the LTP from CA1 to subiculum, so again, the outflowing pathway, you see LTP impairment, but not the incoming, which is the Schaefer. So both systems now show LTP impairment only occurs when you stimulate the axons from the neurons upon which the transgene is expressed, not when you stimulate the axons that's coming in to the neurons where the transgene is expressed, okay? So this means that expression of APP in presynaptic neurons cause synaptic impairments in postsynaptic neurons. It's an anterograde phenomenon. It's what we can delineate using these specifically targeted mice. Okay, is this how synaptic dysfunction propagates? It's interesting to speculate, but of course in brain, since APP expresses pretty much pan-neuronally, we would never see this, but only in isolation, as I just showed you. Okay, what about the synapses themselves? I've shown you measurements by LTP and by the amyloid deposits. What if you count the synapses? And I have an outstanding graduate students who went ahead and started counting synapses in the fields that we would have predicted is where neurons should be affected. So here, just highlighting where everything is, CA3 projects to CA1, and there's a stratum oriens on the outside, stratiatum on the inside. The black here is interesting because the black is supplied from the dentate gyrus, okay? So if you look at these dendritic terminals, they do not originate from inside the hippocampus. CA3 projects to CA1, CA1 projects to subiculum, CA2 projects primarily to this distal arm of the CA1 region. So you can't really see it in the back, and I'm not going to talk about it. Just the synaptophysin will stain puncta that we counted as if they are a single synapse. And then so here at 6 months old animals, nothing happens. Synaptophysin between control and transgenic mice, okay? But if you wait until 12 months, then there's small but statistically significant decrease in synapse counts by synaptophysin in CA1 region in the CA3 mice. That means from here, projecting over to stratum oriens, stratum radiatum, there's a subtle about 10-15% decrease in synapses in the area that we saw amyloid plaques, and we would predict that there should be some effect on the synapses, okay, but not in itself. CA3 is notable for sending recurrent collaterals, that is to send axon projections, not up there, but then come back and synapse on itself. And when it synapses on itself, we can count those synapses, and then we don't see any difference. Okay? At 18 months, we wait 18 and a half months, the same reduction occurs up there, but now we actually see a little bit of a reduction of synapses from these presumably recurrent collaterals, okay? Again, it's a distal phenomenon. And then I didn't quite trust all the data, so we did, instead of synaptophysin, we counted PSD95. It's a presynaptic marker, this is a postsynaptic marker, and we saw the same data almost identically as we saw in the synaptophysin staining. So again, a very nice, consistent demonstration of distal synapse loss that presumably is caused by the A beta that's generated from CA1 neurons themselves. So what about by EM? Let's go one step further. So this is serial block place electron microscopy. So the sections, section one by one, and you can see here one section, and then they are laboriously reconstructed from one section to the next, and we can count them. We can count the synapses to see what happens. And when we do that, it turns out we actually saw greater amounts of decrease in synapses. So that's very satisfying that if you take something more sensitive, which is electron microscopy, you will see more synapse loss in these animals. And then finally, you can reconstruct them by 3D reconstruction on the actual dendritic shaft. That is to say, you take this, you take the next section and the next section, computer will generate out and connect for you what the dendrite looks like, a dendritic shaft. And when you do that, you can count the synapses. And when you see this is a transgenic animal and a control, in that case, in the reconstruction, we actually see 50% dropout in dendritic spines, to even more than what we had counted previously. Okay, and in fact, the residual, the remaining spines are actually a little bigger. And that's been noted by others before, which is initially when there's some synapse loss, the remaining synapses get bigger, presumably as a way to try to maintain synaptic transmission so that you get stronger synapses. Okay, so in summary, in this first vignette is that A beta-induced synaptic injury originates from the presynaptic neurons. Okay, hope I've convinced you of that, and that gene targeting can preferentially target APP to multiple neuronal groups and to delineate the role of pre versus postsynaptic neurons. And impairment in LTP is seen when it's only APP is only expressed in pre but not postsynaptic neurons. A beta is deposited at or near axonal terminals, and then the synaptic deficits and synapse loss are secondary to release of A beta from presynaptic terminals, but affecting the postsynaptic sites. So through all that work, several years of work, we can make these conclusions about these subtle changes that A beta induce directly in mouse brain. So vignette 2, I'm going to change topic and talk about our second model, which is using tau. So almost the exact strategy as vignette 1 instead of APP. Now we use tau. Can I drink some water? Sorry, my mouth is really dry. So in tau rodent model, we have the same thing. First generation, little or no tau or tangle formation when you overexpress just the tau transgene in mice. Second generation, you use neuronal promoter to drive tau expression with mutations that are actually not from AD, but from frontotemporal dementia. There are no mutations in tau for Alzheimer's disease, but they're confined to so-called FTD, frontotemporal dementia cases. And that you saw initially tau pathology that's present, but it's actually much greater in spinal cord than in cortex, and that doesn't happen in human FTD cases. Third generation, again using the TTA regulation, and that's been published by Brad Hyman, Karen Duff's lab. And then the fourth generation, the Rican mice, and unfortunately, if you put the mutation knock-in into the mouse, the tau mutations do not generate any pathology that the authors have noted. So basically, in this particular mouse model, it's not from Karen Duff and Brad Hyman. It's driven by a TTA from the entorhinal cortex, the so-called neuropsin promoter, and just like what I showed earlier, okay? But what we did is here is using the same Cree system that I showed you, but instead of the APP transgene, we put in this transgene with the tau P103L mutation. This mutation, again, importantly, is from FTD, not from Alzheimer's disease, but it does generate tau tangles, just like what we see in the Alzheimer's disease. And this animal line was generated by Karen Chow in Minnesota. And then so here you can barely see it. I will tell you that basically the tau transgene is in red. This is just a little edge artifact, so that's not correct. And you can see some green staining, and that's from the transgene. That's in the tau staining. So here, a better illustration is that in CA3 driven mice, so this is 2 months, 6 months, 12 months old, and then the green down here, which unfortunately you can't see very well in the cell body, are the neurons expressing tau. But beautifully, as the animals get older, is that you can see this band of green staining, and that's where the axonal terminals of the neurons that come out of CA3, they terminate, and then they are all expressing tau. So the green color here is for human tau from the transgene, red is for phospho-tau, which we saw very little, and then the blue is just a DAPI for to stain the neurons, okay? So again, basically similar to what we saw earlier, APP is not abundant in the axons that you could see. We saw mostly staining in the cell body. Here we see staining in the cell body, plus the beautiful localization of tau in the axons, where it does its job as a microtubule stabilizing protein. And then here, a nice picture is worth 1,000 words, so I'll show it again. And you can see how beautifully, here are the tau, the dots for the tau neurons, and then the axonal projections from the CA3 neurons with the tau transgene. Okay? And same thing, we measured LTP first, and you can see that in the transgene animals, there's depression of LTP when you stimulate the Schaefer collateral, just like what I showed you earlier in the APP. Same outcome. But since you can suppress the transgene, if you add doxycycline, then the LTP goes away. So doxycycline will shut down the transgene. And these lovely traces are done by Li Kuei, a talented postdoc in Professor Saji's lab, and he'll be speaking shortly. And so here, just showing that here is a transgenic animals after 4 weeks of doxycycline. You can see that most of the red staining goes away, but there's still some left in the axonal fibers. So the half-life of tau is much longer than that of APP. Okay, same thing you can now record, just as I showed you earlier. You simulate the afferent fibers versus the efferent, which is the outgoing, and you see something quite different. When you stimulate the outflow, just like what I showed you, there's reduction in LTP. When you stimulate the afferent, there's still reduction in LTP. So this is different from the APP setting, which is here. When we stimulate in APP mice, there's no LTP impairment. So tau animals behave very differently. If you stimulate the neurons from the afferent fiber, it would appear that when you overexpress tau in the CA3 neurons, it itself causes a synaptic dendritic dysfunction, which changes the LTP response. So tau expression in CA3 neurons produce distinctly different response on synaptic plasticity than APP expression. So here again, the LTP persists just like the graph that I showed you, and then here it is in the Mouseheimer. Again, this particular tracing is done in Professor Saji's lab, where you can see the loss of LTP, but that is recovered after doxycycline. So take-home message for this second vignette, a little short, which is that A-beta toxicity progresses anterogradely, presumably by release of A-beta from neurons, okay? But tau is different. It shows similar toxicity on postsynaptic neurons like A-beta, but the neurons expressing tau also exhibited impaired function in both presynaptic release. We also saw changes in PPF, that is paired pulse facilitation, that I didn't show you, and synaptic plasticity when afferent fibers are stimulated. And is this why tau may be more toxic and better correlated with better correlated with cognitive impairment in humans? It's been noted that if you have a lot of amyloid deposits in human, it doesn't correlate well with the degree of dementia in the patient, but tau correlates much better. So what I've shown so far is that we have two different models. They're incomplete replicates of AD, but shows different facets of toxicity from A-beta and from tau. So these, as I said in my title, is that all models are bad. It really doesn't replicate AD. It shows certain aspects. And so it turns out Karen Shao, who made that tau animal that we used, had an article which said, title is "Too Much Good News. Are the Alzheimer models telling us how to prevent, not cure Alzheimer's disease?" Because what happens is if here from green to red is from normal to MCI, mild cognitive impairment, to AD stage, all the mouse models is somewhere between a pre-MCI stage. It really does not model AD. Okay, for all the work that we've done from all the labs around the world, these models are incomplete models, which is what I've stressed in the beginning to now. You just have to be very selective as to what questions we try to ask and what these models are capable of answering. They are, as my friend Dale Bredesen, they're not AD models, they are Mouseheimer models. They only replicate certain aspects of AD pathology. How much time? Just want to make sure I have enough time to finish the third vignette, otherwise I won't start it. Okay? The third one is to model now A-beta toxicity using OTSC, that's organotypic slice cultures, okay, to study synapse loss. I've shown you synapse loss from the APP mice. I didn't show you the synapse loss in the tau mice. And then now we want to model them a little more finely. Instead of in mouse tissue directly, it's now mouse tissue that's been dissected from the brain and cultured as so-called organotypic slices instead of dissociated neurons. These are thick 300 micron slices of brain tissue that you can keep them alive for weeks and months if you wanted to, and model certain pathology. In this case, obviously, the pathology we want to model is that of synapse loss, in particular in the dendrites, and using A-beta. So we've known for a long time that A-beta induces synaptic toxicity, and here I call them oligomeric, which means that they're not monomers, they're not fibrils, but they're somewhere in between where they are just multimers of A-beta. And we know that if you give dissociated neuronal cultures A-beta, then you induce LTP and loss of synapses. That same thing happens if you do it in organotypic cultures, which I'm going to tell you shortly. And then finally, that happens in mice, as I showed you earlier, and that those synapse loss recover when you remove the A beta from the mice. Okay. This is a classic study from Dominic Walsh that was published in Nature in 2002, where basically he added A beta that's generated from these cultured cells called 7PA2 from Chinese hamster ovary cells, CHO cells, and when added to acute hippocampal slices, then there's depression of A beta. You only have to treat A beta for about 20 minutes and you stimulate the cells. Here's the LTP, and there's decrement in LTP in the hippocampal slice cultures. And that's due to A beta, as showed elegantly in his paper. But we know this is an example of LTP depression induced by A beta. Okay. Subsequently, a few years later, from Robert Malino's lab, initially in Cold Spring Harbor, before he moved to UCSD to become my colleague, instead of adding the A beta to the culture, he infected neurons in organotypic slice cultures with the Synbis virus, overexpressing APP. When he did that with APP, this is a GFP control, these are sparse infection, so only single neurons are infected at a time. So the green color nicely demonstrates how you can just interrogate only a single neuron, and the dendrites that come from that single neuron. So these are dendrites. And from where you are, you cannot see the dendritic spines. You can see a few here, okay? What happens if you overexpress APP? There's about a 40%, 50% dendritic spine loss compared to control. But that if you put in an APP transgene where you cannot make A beta, I won't go into the details, where you can have no A beta produced, then the spines are not lost, showing that this is a beta specific effect. And then another aspect of APP that I'm going to introduce now is that it's been shown by multiple labs in the same time, this is back in 1999, is that APP is one of many substrates that are keyed by caspases. Caspases are these so-called executioners that carry out the programmed cell death pathway in development, in apoptosis, to regulate a normal body cell turnover. Okay, and it turns out APP is one of many substrates that can be cleaved when caspases are activated. And so what it's been shown is that this VEVD, which is a consensus caspase cleavage site in the cytoplasmic domain of APP, which is a single type 1 transmembrane spanning molecule. These are the multiple cleavages that generate A beta, okay? And that this caspase cleavage is in the cytoplasmic domain and has nothing to do with the A beta itself. That was shown by Nicholson in that paper here and by multiple labs simultaneously. And we showed that as well with Dale Bredesen, but instead of focusing on the cleavage sites, we said that, in fact, after cleavage, something else happens, which is that from the cleavage that happens after this aspartate residue at D664, there's a peptide of 31 amino acids. And we generated data and proposed that C31 by itself is yet another toxic molecule. What's important here is that if you make a mutation so that the aspartate is now substituted for by or replaced by alanine, then APP cannot be cleaved by substrates, by caspases. Caspases have a specific requirement for aspartate residue where it's cleaved. So the D664A is very important. I'm going to talk about it a lot from here until the end, saying that that will abrogate, it will prevent APP from being cleaved. Okay? So this is just a load of data to show that release of C31 is toxic, but it also requires APP itself. So there are two phenomena to note in this slide here, which is that one, in these cells, B103 neuroblastoma cells, they have no endogenous APP through a quirk of this mutant cell line. So now if you overexpress APP, you have some cell death, you have some toxicity. But C31 itself, which you overexpress, has no toxicity in these cells. But when you put in APP, you have APP and you add C31, then you see the cytotoxicity, okay? But if you overexpress D664A and you add C31, there is no cytotoxicity. That shows you there's a phenomenon of C31 toxicity that requires that caspase cleavage site. That's how I showed you this D664A is very important. But that the same thing over here, which is that the A beta toxicity also depends on the APP, and it also depends on D664A. So which is that there are two phenomena going on at the same time. APP gets cleaved by caspases, and if you block that caspase cleavage, you blunt, you lower the cell death response. But that A beta also feeds in, so A beta has an APP dependent component, okay? So now we generated the APP non-kin mice, nothing much to tell you about. We just basically went in there with site-directed immunogenesis and made the D664A substitution. And when we did that, just it's confirming the transgene. First, we you can't see from where you are in the back, except it's not significant. This is endogenous APP. This is endogenous A beta. We know that this knock-in mice doesn't change A beta production, just as a control illustration, okay? And then now we made organotypic slice cultures, so you can see each of these green dots is where a Synbis virus expressing APP is introduced into the slice culture, and you can see now beautifully the dendrites coming out from these neurons. But importantly is that if you take these slice cultures and added A beta to them, just like I showed you Dominic Walsh did, but not measuring LTP, but counting synapses, when you do that, what you see is that first, this is the control, but you add the A beta to the cells, then you have about 40% dendritic spine loss, okay, in wild type animals. But if you added it to the D664A mice, which I'm sorry, you can't see in the back what this is, but the red is the knock-in mice, then the toxicity goes away. And this is at 150 picomolar of A beta. You can see it's quite sensitive to low levels of A beta. So this is the picture that Dominic Walsh showed, and now instead of doing organotypic slice culture, we did the same acute hippocampal slices, just like Dominic Walsh did. When you do that, using the same conditioned media, then what happens is that here you add it to the slices. What he showed here, we have replicated the data, but now if you add the media with A beta in it to the D664A, then the toxicity goes away. So this is a good in vivo demonstration of that toxicity that I showed you earlier in cultured cells, but this is actually in neuronal slice cultures from these mice itself. So there's no overexpression of APP. So now I'm going to change and finish off the last part of my talk, which is I've shown you we made the mouse. I've shown you the rationale for making the mouse, which is the caspase cleavage of APP. And then finally is we want to study, again, the individual dendritic spines after you you deliver A beta to the slices. But we don't want to just add A beta to the media. We want to do what Robert Malinow did, which is only have single neurons express an APP transgene that would deliver the A beta. So we know the A beta is only released from that neuron. And the way we did that is just by a trick, which is making a very short APP construct that has A beta in it. It's been deleted to the C-terminus, so we don't have an issue about the caspase site. And that's just a source of A beta, and you don't have to worry about the details of what we express. It's just that here's a control setting. You put in this transgene, again using Synbis virus, so you only infect very few neurons, and then you see about a 40% drop in dendritic spines overnight. Seems like there is a floor effect. You know, most of the time you only see about 40 to 50% dropout in these dendritic spines, okay? But that now, if you add in caspase inhibitors, remember the D664A site is to inhibit caspases. So there are other ways to inhibit caspase, which is just use the inhibitors. And when you do that with ZFAD, oh, the light just went out, but I can see it. You can still see the screen. If you add ZFAD, then which is a pan-caspase inhibitor, then the dendritic spine loss went away. So we now show that there's a caspase-dependent step, but instead of now using caspase inhibitor, we look at the D664A mice. So same thing, control, setting, you treat it with this construct to deliver A-beta to slices. You see about 50% dropout. But if instead of delivering it to control slices, you deliver it to these D664A mice, then it recovers and it's the same as control. So genetically, we can change one particular amino acid residue in the APP gene, and we can block this A-beta-induced toxicity. So again, it's coming from something related to this caspase cleavage site. So I'm going to end with this last experiment, which is to say we can show that using a bit of genetic trickery to deliver A-beta specific to single neurons, we see that it's dependent on caspases. So how does caspase work? How does it all relate to dendritic spine loss? And what we did here is to use a so-called FLICA reporter, which is a fluorescent inhibitor of caspase activations, something you can just buy commercially. And what happens is that normally when you deliver A-beta through the strand gene, you actually see the green color signal, which is from this FLICA reporter specific to caspase 3, and you see it inside dendritic spines. You see it in the spines that will presumably become lost over time. You don't see it in control, and then you only see it when you overexpress, deliver this A-beta construct to express it in single neurons. The red is just another fluorescent marker to show where we should be counting the cells, that these are the cells that are overexpressing it. And then now if you do a graph to look at spine loss, okay, which is here in blue, and then caspase activation, which is in red, and this is over time, and then just this dew plot showing the red positivity, and then the dendritic spines in blue, you can see very nicely the time correlation. Spines initially are normal at 12 hours, it drops a bit at 18, drops a bit more at 24 hours, and then the caspase activation signal is in the dendritic spine head, and it goes up with time, just as it reverses and crosses over. Okay, so very nice negative time correlation between the loss of synapses and caspase activation. But note that the caspase activation is really only in the dendritic spines locally and not all over the place. And that's important. So then, okay, this is just merging it and showing that now, instead of showing normal spine cultures from normal mice, we do it in D664A mice, the knock-in mice that I told you earlier. Then what happens is that caspase activation signal is gone. So when you don't have spine loss, you also do not have caspase activation. Again, putting those two together. I can't tell you which comes first. I presume it's the caspase activation. Okay, then it's the story that we always keep asking, well, what's ahead of the caspase activation? What's upstream of that? And at this point, I do not know. Okay, so in summary, finishing my whole talk here, which is that there's an APP-dependent pathway of A-beta toxicity that may involve caspase cleavage. And so one pathway of A-beta toxicity is APP-dependent, and which may be secondary to caspase-mediated cleavage of APP. The mutation of caspase cleavage site, that is at position 664, is neuroprotective, and it also reduces caspase activation. And there may be local caspase activation that contributes to synaptic injury. And then the questions that we have to tackle are what activates the caspases in the spines? And finally, how does caspase activation itself cause the local spine loss? Okay. So that's the conclusion for my third vignette, and I want to thank all the work that people have done. The projects from all three vignettes, some were started in UCSD in San Diego, and former and past members at UCSD, at NUS in Singapore. These are the people in the lab. And critically, these are my great collaborators at UCSD that helped us with the EM and the electrophysiology. But then critically, all that work on the tau electrophysiology is supported by Professor Saiji and Li Kui. So thank you very much for your attention, and of course, thank you for the invitation and the honor to come speak here. Thank you.